Slow motion shots of a knife through a tomato, steam rising off a teapot, a square of dark chocolate snapping in half, shot like a food film, not a lecture. VO, cancer cells have a weakness. It's not a weakness in their DNA, and it's not a weakness in their ability to divide. In fact, that's exactly what makes them so dangerous. Their weakness is logistical. They can't grow past the size of a pinhead without their own private blood supply. That single fact has quietly reshaped modern medicine. And it's also opened up a genuinely fascinating area of nutrition research. Foods containing natural compounds that appear to interfere with exactly the process tumors depend on to get that blood supply in the first place. Today we're going deep, food by food, into what the research actually shows, what it doesn't show, how to prepare these foods to get the most benefit, and where the line is between promising science and marketing hype. If you've seen my video on angiogenesis, this is the practical follow-up you've been waiting for. If you haven't, don't worry. I'll bring you up to speed in the next 60 seconds simplified animation. A small dormant cell cluster sitting still, then suddenly sprouting vessel tendrils and growing rapidly once connected. VO. Here's the core idea in plain terms. Most adults by middle age are walking around with microscopic clusters of abnormal cells somewhere in their body. This isn't a scare statistic. It's a well-documented finding from autopsy research. The vast majority of these clusters never become a health problem because they remain dormant, trapped, starved of the nutrients and oxygen they'd need to actually grow. The reason they stay starved is that they don't have their own blood vessels feeding them. And the reason that matters is that a tumor, unlike normal tissue, which already has an established blood supply, has to actively build new vessels from scratch in order to grow into something dangerous. That process is called angiogenesis, and it's triggered by signaling proteins that abnormal cells release to essentially recruit nearby blood vessels to grow toward them. This reframes cancer prevention in a useful way. The goal isn't necessarily never have a single abnormal cell. That may be biologically unrealistic for most adults over time. The more actionable goal is influencing whether those dormant clusters stay dormant by supporting your body's natural ability to regulate that vessel recruitment process. This is exactly where today's foods come in, not as cures, not as treatments, but as low-risk dietary tools that the available research suggests may help tip that internal regulation in your favor. On-screen text. Not a cure, not a treatment, a risk reduction tool built on real biology. A simple pyramid graphic, cell studies at the base, animal studies in the middle, human clinical trials at the top, getting narrower as it rises. VO. Before we get into the specific foods, I want to set expectations properly, because this is exactly the kind of topic that gets wildly oversold online. Most of what we know about specific foods and angiogenesis comes from cell studies. Researchers exposing cancer cells or blood vessel cells in a lab dish to a particular compound and measuring what happens. This is useful, inexpensive, fast. And it's also the weakest tier of evidence because a dish of cells doesn't capture digestion, metabolism, dosage in a living body, or how compounds interact with everything else you eat. The next tier is animal studies, usually mice, often with human cancer cells implanted, then fed diets enriched with a particular compound to see if tumor growth or vessel density changes compared to untreated animals. This is stronger evidence, but mice are not small humans, and dosages used in these studies are sometimes far higher than what's realistic through diet alone. The strongest tier, large human clinical trials, is frankly where this specific research is thinnest. Funding bodies and pharmaceutical companies have far less incentive to fund a multi-year trial on green tea than on a patentable drug, so we mostly have to rely on population studies. Large groups of people tracked over years, looking at correlations between dietary patterns and disease outcomes. Correlational data is valuable, but it can't fully isolate cause and effect the way a controlled trial can. So here's the honest framing for everything coming up. This is biologically plausible, frequently observed in lab and population research, generally very low risk to act on, and reasonably exciting science. but it is not the same tier of evidence as an approved cancer drug, and no food on this list should be treated as a substitute for medical screening or treatment. Here's a concrete way to picture this hierarchy in action. Imagine a single compound, say EGCG from green tea, moving through the research pipeline. It starts in a lab dish where it's tested against tomatoes being roasted, then crushed into a rich red sauce simmering in a pan. VO. Tomatoes owe their anti-angiogenic reputation primarily to lycopene, the carotenoid pigment responsible for their deep red color. In laboratory studies, lycopene has been shown to interfere with several of the signaling pathways tumors use to recruit new blood vessels, and population studies have repeatedly found associations between higher lycopene intake and lower risk for several cancers, prostate cancer being the most studied example. Here's the detail that surprises most people. Cooking tomatoes doesn't destroy this benefit, it enhances it. Lycopene is bound up inside the tomato's cell walls in its raw form, and heat breaks those structures down, releasing more lycopene in a form your digestive system can actually absorb. Adding a small amount of fat, olive oil for instance, further improves absorption since lycopene is fat-soluble. This is part of why a simmered tomato sauce with olive oil is, from a lycopene availability standpoint, a meaningfully better source than a fresh tomato salad, even though both are healthy in a general sense. Practical takeaway. This doesn't mean raw tomatoes are bad for you. They offer plenty of other nutritional value. But if you're optimizing specifically for this mechanism, cook tomato dishes a few times a week, think pasta sauce, roasted tomatoes, or a simple shakshuka, are the more strategic choice. Green tea leaves steeping in clear glass, steam rising, golden green color developing. VO. Green tea is arguably the single most studied food in this entire field. Its key compound, EGCG, short for epigallocatechin galat, has been examined in hundreds of laboratory studies for its ability to interfere with the specific growth factor signaling that tumors use to trigger new vessel growth. What's notable about green tea specifically, compared to black tea or oolong, is processing. Green tea leaves are minimally oxidized during processing, which preserves a much higher concentration of EGCG compared to teas that undergo more extensive oxidation. This is purely a processing difference, not a difference in the plant itself. Green tea, black tea, and oolong all come from the same plant species. Brewing method actually matters quite a bit here. Water that's too hot, boiling for instance, can degrade some of the beneficial catechins, while water that's too cool won't extract them efficiently. Most research-aligned recommendations suggest water around 175 to 185 degrees Fahrenheit, roughly 80 to 85 degrees Celsius, steep for 2 to 3 minutes. Steeping much longer doesn't necessarily mean more benefit. It mostly means more bitterness, since tannins extract more slowly than catechins. One genuine point of nuance. Most human studies on green tea and cancer risk are observational, looking at populations with high habitual green tea consumption, particularly in parts of East Asia, and comparing cancer rates to lower consumption populations. These studies show encouraging associations, especially for certain digestive cancers, but they can't fully rule out other lifestyle and dietary differences between populations. Still, given how low-risk regular green tea consumption is for most people, it's one of the easier dietary changes to justify on the strength of the available evidence, a bar of dark chocolate being snapped, cocoa powder being measured. VO. This is everyone's favorite one to hear about, so let's be precise about what the evidence actually supports. The benefit here comes from cocoa flavonoids, plant compounds concentrated in the cocoa solids, not in milk, sugar, or other additives. Research on cocoa flavonoids has primarily focused on cardiovascular benefits, improved blood vessel function, modestly better blood pressure regulation, and some of this research connects to angiogenesis-related vascular signaling specifically, since healthy blood vessel function and angiogenic regulation are closely related systems. The critical detail: benefit scales with cocoa content, not with how indulgent the chocolate tastes. A chocolate bar that's 70% cocoa solids or higher contains meaningfully more flavonoids than a 30 to 40% milk chocolate bar. And the added milk and sugar in lower cocoa products can blunt some of the absorption benefits, while also adding the kind of added sugar that works against cardiovascular health in other ways. So this isn't really chocolate as healthy. It's specifically concentrated cocoa flavonoids, found mainly in high percentage dark chocolate, have demonstrated vascular benefits in research. A reasonable, evidence-aligned approach is a small portion, roughly 1 to 2 squares, of 70% plus dark chocolate a few times a week, treated as a flavorful addition rather than a health supplement. Eating an entire family-sized dark chocolate bar daily isn't going to scale the benefit linearly, and the calorie and fat content adds up quickly at that volume. Overhead shot of mixed berries, strawberries, blueberries, raspberries, in a bowl. VO. Berries bring two major compound families into this conversation. Anthocyanins, the pigments responsible for their deep red, blue, and purple colors, and ellagic acid, particularly concentrated in strawberries and raspberries. In cell studies, both compound families have shown the ability to interfere with angiogenic signaling, and some animal studies using cancer models have shown reduced tumor growth and vessel density in groups fed berry-enriched diets compared to control groups. Strawberries specifically have drawn research interest for breast and cervical cancer cell lines, while blueberries have shown effects in studies on several different cancer cell types. A practical note, frozen berries are not a downgrade here. They're typically picked and flash frozen at peak ripeness, which can actually preserve more of these compounds compared to fresh berries that travel long distances and sit on shelves for days, slowly degrading. If cost or seasonality is a concern, frozen berries are a fully legitimate, evidence-aligned substitute. Garlic cloves being crushed with the flat of a knife, releasing visible aromatic compounds. Illustrated, not literal. VO. Garlic's relevant compound is allicin, which is actually formed only after a garlic clove is crushed, chopped, or chewed. Raw, whole garlic doesn't contain allicin itself, but rather a precursor compound that converts to allicin on contact with the enzyme released through cutting or crushing. This matters for how you prepare it. Allicin is also somewhat heat sensitive, so if you're cooking with garlic and want to preserve more of this specific compound, crushing the garlic and letting it sit for several minutes before adding it to heat allows more allicin to form first. Garlic powder and long-cooked garlic, like a clove simmered for an hour in a stew, will retain meaningfully less allicin than fresh garlic added toward the end of cooking, or eaten raw in something like a dressing. Research on garlic and cancer risk includes both laboratory studies on allicin's anti-angiogenic activity and large population studies, particularly out of regions with traditionally high garlic consumption, showing associations between garlic intake and lower rates of certain digestive cancers. As with green tea, these population studies can't fully isolate garlic from other dietary and lifestyle factors, but the laboratory mechanism gives the correlation a plausible biological backstory.